All right, so yeah, the title is, it should just be pre-processing. And I'll be focusing on the first class of methods that were introduced, the imaging-based, and we'll hear about the sequencing-based, I think, tomorrow or the day after. And I wanted to start with a picture that this is my data at the moment, and I want to start with a bit of motivation of why we care a lot about quality control and processing. So before we get our data on a screen as a computational biologist, this has maybe been sitting in a hospital, a biobank. They have hundreds of samples that have been diagnosed, treated. Maybe the people died, maybe they went into remission. And now we want to do science, and they will select maybe some interesting cases. Or we're interested in development, and we section brain at different times in development. All right, so then someone has to do a cut. Then maybe this sits in storage. It's fixed. The mRNA deteriorates. It sits there for a decade. Then in this case, they send it to Seattle, and some liquid gets into the box, and it gets destroyed, right? And then at the end, we run this through either a sequencing facility, or we have a machine in the basement where our wet lab of PhD students has to handle that and run that experiment. And then in the end, we get our data. And then what we're doing then in R is we have this data, but before that, a lot of things can kind of go wrong. So kind of to schematize that, we have a tissue. Typically in pathology, we'll have like an H&E staining, and a lot of pathology can be inferred from that by experts who've been doing pathology for decades, right? Or by AI-based approaches. On the other side, now we do imaging. We do an immunofluorescence staining maybe. We do high-plex FISH, so then we get all of these molecules, as was shown in the previous lecture. We do segmentation, we get these boundaries, and then we can count the transcripts, and then that's the analysis that we in the end want to do. And the issue then, or the challenge a bit, is that this is already pretty complex compared to just single-cell sequencing maybe. Each of these then comes with a different, let's say, data type. So we have images. The high-plex FISH are also images of individual spots. An algorithm has to call the mRNA. The segmentation is an algorithm. We get polygon boundaries. We have points. And in the end, we have a count matrix. So each of these are also different views of the data in a way. And I want to today go through some of these. And maybe to point out a bit of the challenge here is that in single-cell RNA-seq, I mean, we're a bit more advanced now, but in the beginning, we kind of know the process. We know we have fragmentation, reverse transcription, we map to a reference genome, we get our counts. We know that that's affected by GC content and blah, 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 and there's PCR amplification biases. So we know all of that process, and then we kind of know how to deal with that. So we know we should do normalization, we know kind of what we do with that. In imaging, we kind of have a similar story. Our tissue is being prepared, we have different chemistry, we have imaging. Tissue can get damaged, transcripts can be lost. But in this paper, they kind of then say, although we kind of know that, we don't know how it occurs, and we don't really know how to deal with it. So sort of as a disclaimer for my slides, at least, is that we don't really know what's best. And a lot of the slides I'm showing are papers from the last couple of years, and they're more like ideas of how people are trying to deal with that. After, I think, for 5 years, people have been publishing vast atlases, and they haven't really dealt with the problems in the data. And there's been less work in kind of the early stages of the analysis. Okay. So one of the first things we can do is forget about the cells and forget about the count matrix. We have images like a DAPI staining, and we have our transcripts, and we can do quality control and look into things at this level. So before we even have a count matrix, just considering our transcripts. And there's this method that called Merkrow, I don't know how to pronounce it. One thing they propose, which I thought was quite neat, they look at the transcript locations and the DAPI staining to train a classifier to basically create different kind of masks. So one is like a transcript mask that tell us which pixels here have transcripts. One is a DAPI mask. We can subtract the DAPI from the transcript to get like a detachment masks. And I'm cutting off some more complicated combinations of these where they create binary masks on the tissue. And by combining these, they can classify the pixels in the image. And what they then get, and I mean, it's quite intuitive in a way. We have a tissue where we kind of then say, okay, what's tissue, what's damage, what's detachment, and what's really out of tissue. And this is a very early stage before we've done any computational analyses where we kind of say which regions of the tissue are already maybe problematic. In this paper, they also propose a bunch of other metrics, and I'm not going to go through all of them. I just want to mention them. So one is like the machine usually gives you a log file where you can look at if the flow kind of was continuous or if there was any blockage. If there is data loss, so by comparing every pixel or field of view to its neighbors, you can see if there's really bad locations in your tissue. Then the detection efficiency, one across the section and the other across the Z stack. So in like 3 dimensions, if there's differences. And finally, the transcript density. And I just want to show you one example here where they say, if we just count the total number, we do a histogram of the transcripts from left to right and from top to bottom. Of course, this will depend on the orientation of that section. So here it's really nice. And then here it's different because this is like tilted. But if you had consecutive sections and they're registered, it should look the same, more or less. And then they say, okay, for the first one, it's pretty good. But for the second one, there's like it's much lower. And there's like a drop. So something's weird. Similarly here, this is the P6 to P0 ratio. So that's the Z stack. So again, these are 3 Z coordinates. And then they kind of say this one is pretty stable. But in this one, here, basically, we don't really get any transcripts anymore. So that's really bad. Yeah. I thought this was quite interesting. I don't know yet how to apply all of these things because it's very applied to their data. But I think the ideas that they propose are quite interesting. Okay. Now, so that was like this part. On the other hand, we can go to the cell level. And for that, as has already been mentioned, we usually have a composite of a bunch of IF markers. We do segmentation. We count the transcripts to get a count matrix. And what we then get is more than just a matrix. Sorry, it's a bit small. For every cell, we get XY coordinates. This is typically the centroid of the cell. And for every gene, we get a stack of X and Y coordinates for the transcripts or the targets that we're targeting. And in addition, we have one sort of vector you can think of as the extracellular targets. So these were just not assigned to any cell, but they're still part of the data set. Like they still exist. Now, once we have our count matrix, we then want to do analysis. And one of the first thing we would typically do in single-cell RNA sequencing with our counts is we would look at cells that don't have any counts or cells that have high mitochondrial content, and we would do these kind of QC metrics. Now, in specifically the targeted approaches, I think there's a lot of issues with those classical quality control metrics. And I just did here like a very basic example. This is base R plots of a random data set that I picked, where we have here the number of transcripts on a log scale, and here we have the cell areas. So this is pretty nice. The cell areas usually have a really nice normal distribution. It really depends on your tissue, of course. And then the transcripts are kind of like this. And then typically what we would do is maybe in single-cell RNA-seq, we take some cutoff here and remove cells that are below that, right? But we know that the bigger the cell, the more transcripts it should have, right? So we can look at something like the number of transcripts per pixel. And then the shape is a bit weird now. So there's a little bump over here. And that's kind of it's not a perfect fit. There's even data sets where you'll find a little cloud up here or a complete shift of two clouds, so it's not a perfectly linear relationship. And if we take away the log, this is now the absolute number of transcripts per pixel. Again, we find this little bump here is a bit more visible. So this looks a lot like a mixture of two different things. So a part of the tissue has just more counts per pixel than the other, which, I mean, it makes a lot of sense, but I feel like lots of people ignore it. So what I did now in the next plot, what you will see is, okay, let's say I do QC, I set a cutoff here, I set a cutoff here. That would be like setting a vertical and a horizontal cutoff. Alternatively, I could set a cutoff here, and that would be like setting a diagonal cutoff, right? So all of these are legit things that I see people do, and I also do that for lack of a better solution. But then what happens downstream? I analyze my data, I annotate my cells, I look at it in the tissue, and then I see here the epithelial cells tend to be here, and the immune cells and the stromal cells tend to be here. So I've basically excluded a bunch of immune and stromal cells. And my epithelial cells, they're just, bigger and they have more counts, and I'm not filtering on those. And it can be so bad that the biologist will ask you what happened to the immune cells or what happened to this, like it's gone, it's just gone, right? You removed it. So just to show you a couple more examples, this is again the same colors as before. We can look at the DAPI state, the different IF markers. We can look at the cells over the tissue. We can look at distributions of things. But what I would really recommend people do is also to look at that downstream or to look at it throughout the analyses. Because what you will find this is sort of a compartment level view. At the cell type level view, it's even worse. So what you kind of get is, let me pick a good one. Granulocytes are kind of known to be, maybe transcriptionally less complex, and they're tiny. Neutrophils are another candidate for cells that we lose a lot. So all of these things are, biased against specific subpopulations. So what I'm thinking now is it better to be lenient in filtering cells and doing it more downstream? Because we're really not sure upfront what we're killing. And the main thing then is to look all of this in space. Since there's a spatial data, we don't want to just look at distributions. And that's an example. I'm really sorry, it's hard to see. But what I typically would then do is I look at my tissue and I just highlight the cells that I've been removing. And if you do a bad job on this, you will see clusters of cells, perhaps. Sometimes there's a whole section of the tissue that's removed. Then it's important to check maybe with a pathologist, is there something wrong? Maybe that's, necrosis. Maybe this part of the tissue has been detached, and there's a legit reason. In this case, that in here is mostly smooth muscle and myofibroblasts, so it makes sense we're like removing a lot of cells. And then actually for this tissue, I emailed Brooker and I asked them about this field of view. And then they said this was out of focus. The microscope didn't get that field of view. So now it's been removed. So just, be kind of sure that what you're seeing with the filtering makes sense. And if it doesn't make sense, you should rethink maybe. So I want to just throw one thing out there that I'm not sure we've heard about in the previous lecture. It's field of views, which I think maybe everyone in this room have heard of. That what I was skipping over a bit is that what we do in reality is we have an H&E staining, and then in Cosmex, you place these field of views. In Xenium, I believe you don't. I think it just scans the whole thing. But in Cosmex, this is what you do. You place like little red boxes. And then you do your imaging and you do your analysis and you're fine. But what then happens is in reality, the performance on the edges deteriorates. So this is I chose this because it's a particularly bad sample. It's particularly bad data. where you see these boxes where on the edges, the upper right corners are just super dim. That's because each field of view was separately imaged, right? Again and again and again. And there was this blog post that wrote, "Cosmic sucks because of this." But the truth is that that's also the case in Xenium, but they just don't tell you about it. Essentially because here in this paper, they write that we segment every field of view individually. And here they solve this by segmenting continuously, but it still exists. You still get these artifacts. It's just less pronounced, but it's still there. And it's just harder to see. And what you saw earlier is that I'm removing some of the cells that are just right on the border. Yeah. And that's because of basically, yeah, this is the blog post that I was writing about. They show these are two adjacent field of views. That's the same cell. You kind of see it has the same transcript, but then the green one is missing over here, and that one is only observed in this cell. And then they say the issue is we have fragmentation, we have duplication, and they don't have the same counts. They get different kind of transcriptomes on these borders. So one thing that I did in the previous data set is kind of shown here. We can quantify the distance of every cell to the border. So if this is a cell, how far is it to the top, to the right, to the bottom, to the left? And we can plot that against the counts, and we see that very close to the borders, there's this drop. The cells are cut in half. And then what I did in the previous plot is I say, okay, if it's closer than 30 pixels, I remove it. 30 pixels, I mean, that's really tiny. It's like not even a full, it's like a third or a fourth of an actual cell diameter. So it has to be really close to the border for me to remove it. And another thing that you might see is that this sort of looks the same as this, but those are different. And that's what I don't know, I call it like a corner effect. So there seems to be a difference between the top left and the bottom right. And that's what you can kind of see here is that the DAPI and the CD45 staining was a bit dimmer in one of the corners. And you could see that at the count level. And that makes sense because the segmentation relies on the staining. And if the staining is really bad on one side compared to the other, you will get these kind of artifacts. But this machine has been replaced, so I wouldn't say that's a typical kind of thing. All right. Yeah. This is the same plots as before, but what is also shown here is the aspect ratio of the cell. So of course, if we're cutting the cell, it also, on average, might, in the distribution, we get slimmer cells, so they're thinner, and they have fewer counts. And what this should sort of teach us is that we don't want to just remove small cells, we don't want to remove low transcript cells, we don't want to remove these, but we want to take a combination of all of these things as a criterion to deem a cell useless, right? Because just because it's small doesn't make it useless. And that was sort of what, this is currently just a GitHub, but I think it will be a preprint soon, where they kind of take all of these different metrics, counts, area, aspect ratio, and then they say, okay, if we pick outliers based on all of these, we get something, but if we combine them all, we get some kind of score that combines all of these different criteria together. And then we say, okay, these are the actual bad cells based on a combination of quality control metrics. And in that case, that makes a lot of sense because those are not cells. So those are not cells. And sorry, just to convince you that I was happy when I saw this talk from that guy because I had a data set that had exactly this. So this is a cancer section. And there's this bubble down here that was annoying us because we didn't know what it was. This is, again, the bubble. And out here is like epithelia. And in here, there's DAPI, but they're not cells. Like, we really don't know what they are. And the problem is that the segmentation thinks they're cells. So the segmentation sees the DAPI and it segments a bunch of cells. And what we think now is that this is debris that the tumor is flushing out, or it's like dead cells, or it's not clear, but it's debris that is being segmented. And I see this like happening a lot whenever you have like fragmented cells. And then the previous approach would sort of pick up on this and exclude those as being problematic cells. The first thing we would do after our quality control is to then normalize our data, right? We would want to go to something like an expression scale to then do sort of downstream analyses. And I think that's another big problem that people are kind of skipping over in most papers. There was a paper that came out, I think, last year where they kind of show, okay, if you have 100 genes that are skewed towards a specific region, you tend to get very high size factors. And then if you normalize, you get very low expression. If we rephrase that, it's like saying, if you're measuring a bunch of brain genes in the brain, you will observe most of the transcriptome, you're fine. But for your immune cells, you will get like 0 counts, right? Because you're just observing a part of the transcriptome. So your panel design, especially in the low-plex method, will really skew the size factors and in turn the expression, which makes a lot of sense. Still, we're applying whole library size normalization, but the word library in this context doesn't exist. There is no library, right? In a 300-plex kind of panel. And then they show, I mean, it's just observational at this point, but they show if you don't normalize, or if you just normalize by cell volume or area, your differential gene expression results will do a better job than if you normalize the way that we're normalizing. So kind of don't normalize. Not normalizing is better than normalizing the wrong way. They then show, okay, I mean, this is a simulation study. If we then do 50, 100, 500 to over 8,000 genes, this effect becomes less and less dominant, which again makes sense. The more genes we're targeting, the more closer we are to the true transcriptome of the cell, and then this effect is kind of mitigated, right? So this, I would say, it's more problematic for xenium than a 5K plex xenium or whole transcriptome kind of things, right? The disappointment with this paper was then that they just say, okay, if you have cell volume, do that, and if you don't, in the end, do a count-based normalization if you think your panel is representative. I mean, the answer is just like there is no real solution yet. I think people are working on this. But yeah, currently there's not really a good way. After normalization then, what's the next thing we would do? We would do like PCA and then plot a UMAP and then cluster our cells, right? I think that's the biggest thing we would do is cluster the cells. And I just want to show you one of the things that then can happen or happens a lot, I think. And is, yeah, so here's a heat map that I get after I've done some analyses. This is marker genes, and that's our cell type. And what I'm highlighting here is that these are B cells. So you can't read it, but there's some B cells in here. There's CD79 in here. And those are macrophages. So there's like MMP, EPOE, lysozyme in there. Okay, great. But what's this? It's like a cluster that has, it's a CD68 positive B cell cluster. And this is really strange, right? In single cell, you would call this maybe a doublet. But that's also weird that there's a doublet forming between exactly like these two cell types and nothing else. And if you look in space, what you see is that here's blue, DAPI, those are cells, and CD45, which is marking immune cells. And we have these black holes. And there's a bunch of CD19 everywhere because those are B cells. And then there's all of these macrophage genes that are in the black holes. So that's weird. When we saw it, I mean, now it makes a lot of sense, but the first time I saw it was weird. It turns out there's a paper that talks about tingible body macrophages in this tissue that eat B cells. So these are like dendrites. There's macrophages with dendrites that eat the B cells. Okay, that's what's happening here. But if you don't know about this and you only see this, kind of what happens is that the transcripts are bleeding into cells that they don't belong to. So these macrophage genes that are being expressed are being assigned to the B cells after the segmentation. And then we get these weird kind of mixture populations, right? And that's a problem because our clustering thinks this is a separate population, but in reality, those are B cells that are being infected by transcripts from B cells that are eating them. Okay, just to show you another example. So this is exactly the same data set that I was showing you. And if you do a UMAP, you also see the same phenomenon. So this spatial bleeding, because it goes in your counts, it goes in your principal components, it also goes into your UMAP. And here's an example. So it's really nice in this case because so these bubbles are here. That's like germinal centers. This cluster here is the epithelium. It's also very separate, right? And then all of this is like the T cell zone kind of in between the bubbles. But the problem here is that we have this B cell cluster that's completely split into two. And that's really a thing that happens a lot, I think, in spatial data. And that's because this B cell cluster is split between membrane and non-membrane. And if it sits in the membrane, it's being infected by keratins. So then the UMAP puts it over here. Or, I mean, the PCs put it over here. And then these other B cells, they're not infected by keratins, and they end up on a different side of the UMAP. And I think the point I'm trying to make here is that if you don't have this annotated, it's a real pain because you see populations that express genes that shouldn't be there, and it becomes very hard to interpret. So this is, again, the same story, but in a more systematic way. So whatever I just showed you was like things I saw, but then there was a paper, I think it just came out a couple weeks ago, where they show this, where they take single-cell RNA-seq data and spatial data, and they show that there's these artifacts here of markers that shouldn't exist. And then they try to kind of characterize that. So on the one hand, they say this usually, so here in turquoise, we have fibroblast markers, and in red, like malignant markers. And usually it occurs on the sides of the cell, which makes total sense, because usually it's like your neighbor that is infecting you. And then they also show that it also happens if you look at a three-dimensional, this effect in 3 dimensions, it tends to happen on the top and the bottom, which again makes sense because there could be cells sitting on top of you, right? So this spatial bleeding kind of occurs in space around a cell, which makes sense. And then they show, because of this, again, it's similar to before. If we look at like a tissue and we have a stromal compartment that's like fibroblast and endothelium and whatever, and then we have a tumor, and we compare these regions, and then the tumor region has like a lot of malignant cells. Now we do differential expression analysis, which is like the heat map that I showed you earlier. What you get is that the fibroblasts express a lot of keratins. Like that's what the analysis is telling you. So this is exactly the point that was the case earlier, is that essentially your differential expression analysis is being made dirty by the microenvironment of the cell. So whatever the microenvironment of the cell, it will kind of affect your differential expression analysis. And this, what you just said, is then what the current methods that are trying to fix this, they rely on a single cell reference. I mean, or they work better if they have a single cell reference. So one of them, for example, and this is the same paper as the last one that I showed, is basically they say, okay, for every single transcript, we look at the neighborhood of transcripts, and we decompose that. So it's like a non-negative matrix factorization, where the transcript's neighborhood is sort of determined by its neighborhood. and there's some hidden kind of latent factor. And then ideally you take reference single cell profiles, right? That would decompose that. And then essentially what you're trying to do is to cluster the identity of the transcript based on its neighborhood. That's the basic idea behind it. And then, I mean, here's an example where this is a malignant cell. This is, malignant markers and fibroblasts. And this approach would cluster them into five, but three dominant groups. One is over here, one is over here, and one is over here. And basically by then cleaning these, the method would then return you a clean transcriptome where sort of this spatial bleeding, they're removed. And that's sort of the idea that, yeah, I don't know if I explained that well. You can read the paper. Yeah. Sorry, another approach that I'm using, I'm not using this one yet, it just came out last week. But it's this one that was also published by Nanostring Booker. I found this one a little bit more intuitive. So they say, okay, we have a cell that has some cluster label. So we call it like a B cell. We take a reference transcriptome from a single cell data set. And initially, we score all of the transcripts based on that assignment. So if a cell has been assigned as a B cell and we have macrophage counts, we kind of score that cell as being problematic. And for the problematic cells then, we look for transcripts that don't align with the reference. So if our reference has like very clean B cell and macrophage populations, right? We shouldn't be observing mixtures of these two ever. So we first like flag cells, we then flag transcripts that don't belong to the cell, and we can then have different solutions. So either we cut it off and we remove it, or we, like here in this case, we trim the cell. We can create a new cell, and we can say, okay, this part of me is like another cell. Or we can take these transcripts and assign them back to its neighbor. So this all kind of depends on the context. And in the case of this example here, what's going to happen is these cells get flagged because they're dirty. The macrophage-related genes within these cells will get flagged because they don't belong there. And then we will correct the counts of these cells and potentially create a new cell in this black hole. That's what this approach would sort of want to do in this kind of scenario. Yeah. So I just wanted to finish a bit that kind of we've heard about transcripts and images and polygons and counts to talk a bit about how we represent all of these data in our computer. And I think some of these things we will then have in the next couple days. So if you're an R user, which chances are you're not, less and less people are, there was the spatial experiment that sort of extends the single cell experiment class in R. The basic idea is that all of your data is represented as tables. All of these tables are linked to each other, but everything is sort of like tables that have links to each other to allow for like synchronization across the object. And the reason why we want to do that is because there's tons and tons of infrastructure around single cell data. So a lot of the methods from single cell we can also use for spatial transcriptomics. So it's really great when, at least in our computer, they kind of work out of the box by relying on a similar file format. This has since been extended. For example, this is from the lab of the Museum of Spatial Transcriptomics paper. So the same lab proposed kind of this, which is exactly like that. But in addition, it can store geometries, for example, spots on a Visium chip, polygons for segmentation, and that those, or pathological annotations of regions, any sort of like geometries can be stored. Again, as I already said, the issue with all of that is it's all tabular. And I think this is really a mistake in the design because this doesn't really work for spatial transcriptomics. The data is just more complex, especially if you're thinking multimodal. if in the same data set you have Visium, Xenium, and images, it's not easy to align them all in tables anymore. So the Python people came up, I think, with a really fantastic kind of framework where they say, okay, all of these technologies that Lars presented, you can present them all as 5 elements: tables, points, shapes, labels of pixels, and images, which are pixels. And just taking these 5 things and representing them on the computer in like nice file formats, we can cover 30 different technologies. And this is, I mean, a very smart design compared to designing a specific data structure for Visium or for Xenium, to really go back to the roots of what is actually making up these data sets. And what that then allows them to do is really cool. What they show in the paper is they have like Xenium replicates and an H&E, and they can like align all of them in 3 dimensions, even though the H&E is pixels, the Visium is spots, the Xenium will have molecules as well as polygons, and we can align all of that by representing it as these primitive kind of elements. And I think you'll be also using this in one of the practicals. And with that, I'm finished.